Hello everyone. I hope I am audible to all of you. A very good evening to all of you. Welcome to another session of CUET PG Prarambh Series. This is day 12 and topic for today's session would be replication, transcription, translation and DNA repair. This is a very important topic which we are going to discuss because a significant number of questions they are asked from this part in the CUET PG exam, as well as covering this part would help you to cover a substantial portion from CSIR net syllabus as well. Not only that, this is part of GATE syllabus as well. If you would go through the GATE BT exam, you would find that all these topics are included there as well. Okay, so yeah, shall we start with our today's session? All right, so let us start with DNA replication foremost. As you might realize that all these topics are quite wide. Okay, so I would try to focus on all the important topics and terminologies which are very often asked in the exam. Okay, so foremost, what is DNA replication? The process by which DNA makes a copy of itself during cell division, that is known as DNA replication. Right, now we must know that what is the significance of DNA replication, right? Why DNA replication is even needed. So see, whenever a cell divides, the two daughter cells, they must contain the same genetic information. As we know that DNA exists as the genetic material for majority of the organisms. Now, so whenever a cell is going to divide, this is important that the daughter cells, they must also have their own copy of DNA, right? So for that reason, the DNA has to divide. Okay, so it would be making an exact copy of itself so that the DNA copies, they could be distributed to each of the daughter cells being generated during cell division, right? So I hope now you have understood that what is the need of DNA replication. Now next question you could ask that what is the need for cell division? Why when the cells divide? Why do cells divide? Yes, would you like to answer on that? Thank you, Pratik Pradhan. A very good evening. Kaushal Mishra. So I hope my voice is all clear. So I would like to get your answers on my question. Why do you think the cells do divide? So see, the main reasons, right, for which the cells they would be dividing are definitely growth, then for healing and regeneration or repair, and then for reproduction. So for any of these purposes, when the cells would divide, they need the DNA replication to accompany that. Yes. Now, let us have a look over a simple question which has been asked, right, based on DNA replication in the previous exam paper, right? DNA is considered as genetic material because of its. Now, 4 choices have been given. So, yeah, can you please let me know what do you think? What should be the reason that DNA is considered as the genetic material? Means which of the following property of DNA makes it to exist as the genetic material in majority of the living organisms? All the above. All right. Okay. So that's what Shrivani thinks. Can I get few more responses? Yes. What do others think? Which of the property, which of the given properties is most relevant for the DNA to exist as genetic material? Don't you think it should be the first one, autocatalytic? This is the main reason, right? Because DNA has the potential to replicate, to make a duplicate copy of itself. You know, this was the biggest dilemma among the researchers long back, right? When they got to understand the basic structure of DNA. But even then, they were thinking that how this is possible for DNA to make faithful copies of itself. That is how the replication mechanism takes place. Okay. So here comes my second question. This is again a previously asked question, right? So have a look on the question. This is really, really simple. DNA replication in E. coli follows. Yes. So can I get some quick answers in the chat section? DNA replication follows. Yes. Is it semi-conservative, dispersive? Yeah, Shrivani, definitely DNA can mutate, right? But that doesn't make it as the most appropriate molecule to exist as the genetic material, right? Means any molecule which should be able, which should act as the hereditary material, foremost, it should have this property to replicate itself, to make duplicate copies of itself, so that whenever the cells divide, each daughter cell that should have its own copy. Okay. Yes, Shivani, that is correct. Semi-conservative model, that is the right answer. Now, based on this question only, I am bringing my next slide where we are going to discuss 3 possible modes of DNA application. Right. Now, Watson and Crick, they proposed the structure of the DNA, right? We know that. They also suggested the semi-conservative mechanism for DNA replication, accompanying their proposal for the structure of DNA. Right. However, due to lack of evidence on the same, there were other theories as well, right, going in the minds of the researchers. So besides semi-conservative, you should have an idea that what do we mean by conservative or dispersive mode of DNA replication, right? But even before that, we must learn about semi-conservative, because definitely this is the mode of DNA replication which is actually being followed. So what happens? DNA is a double helical structure, right? As per the structure revealed by Watson and Crick, we know that DNA is a double helical structure which is made up of 2 anti-parallel running strands. Now, both of these strands, they unwind, just like we open up the 2 sections of zipper, okay? 2 halves of the zipper, they could be separated. So these 2 strands of the DNA, they could be separated, and each strand that would be acting as the template, okay, and complementary to it, the new DNA strand that would be synthesized. As per the semi-conservative mode of DNA replication, the daughter DNA duplex that would be made up of one DNA strand which is derived from the parent, and the other one which is the newly synthesized. So here you would find that the hybrid daughter DNA duplex that could be seen, right? However, in conservative mode, what was thought by the researchers at that time, that there is another possibility, and the possibility is that maybe the 2 parent strands, they reunite to produce the new daughter DNA duplex. And the newly synthesized DNA strands, they are coming together to generate a daughter DNA duplex which is entirely new. As you could see that this is not having any of the strands in blue color, right? Both of them, they are orange. So this is depicting that these are newly synthesized strands. Whereas in the dispersive, right, what dispersive mode suggests? It suggests that DNA strands, they break up into fragments. Okay, and then each of the fragment that may undergo replication, and when it comes to reunion, so the DNA fragments randomly they reunite, right? So some of them, they are parental, as you could see that some of them, they are blue in color, the others are orange in color. Okay, so again these are hybrids, but these hybrids are different from what we are looking in the semi-conservative. So these are the 3 modes of DNA replication which were thought as the possibilities by the researchers when they were trying to track the DNA replication mechanism. So I hope this is all clear for everyone. This is really important to understand, right? That what were the suggested modes of DNA replication. Now, as we know that semi-conservative mode is the actual mode which is followed by the DNA replication mechanism. That was proven by Meselson and Stahl experiment. So the next part which you need to cover in replication topic is Meselson and Stahl experiment. This was considered as the most important experiment because it gave the evidence to support the semi-conservative mode of DNA replication. Okay, now to determine that which among these three modes actually exist, Meselson-Stahl, they wanted to track the DNA, right? Now for that, they labeled the DNA, right? And for labeling purpose, they used the heavy isotope of nitrogen, right? They wanted to track that where the old DNA is going or where the new DNA is being synthesized and how they are reuniting. So for that they need to track the DNA, right? And they label the DNA with the heavier isotope of nitrogen, and that was nitrogen 15. Okay, so this is against the use of the regular isotope of nitrogen, which is nitrogen 14. So what they did, they grew the bacteria in the media with all heavier nitrogen only, right? So obviously when the bacteria would be having only heavier nitrogen, so the DNA which would be synthesized during that phase, that would be incorporated with the heavier nitrogen form only. So that DNA produced during that phase, that is known as the heavier DNA or heavy DNA, right? Now, later on, they shifted the bacteria to the regular media. Regular media means, again, they exposed the bacteria to the regular form of the nitrogen, that is nitrogen 14. So, again, the bacteria started using the regular nitrogen. So, why Meselson-Stahl did this? Like I have told you, they wanted to track or they wanted to differentiate between the older and the newer DNA, right? Now, the next step was, all right, they have labeled the DNA. Now, they wanted to track the movement of the DNA, okay? Or we can say that they wanted to distinguish between the older and the newer DNA. So, how they are going to do that? How they are going to find out that where is the heavier DNA and where is the lighter DNA? So, for that, they use the equilibrium centrifugation method, right? They particularly used cesium chloride density gradient centrifugation method. Now, what happened in this or under this method? Cesium chloride, this solution, this is subjected to centrifugation at high speed, right? So this produces a linear gradient. Linear gradient, what do we mean by linear gradient? Means layering, right? So distinct layers they would be produced. Gradient means layers, and these layers would be produced based on the density, right? That means the layer which would be the heaviest, that would be lying towards the bottom of the tube, whereas the one which would be lighter, that would be towards the top of the tube. Now, when this solution, it is mixed with the DNA, again in the DNA we have mixture of heavier and the lighter DNA. So the DNA that would get equilibrated, that would come at an equilibrium at a point where its density would be matching up with the surrounding density of the cesium chloride solution, right? So that means we'll have the heavier DNA or the heavy DNA towards the bottom of the tube, whereas the light DNA towards the top of the tube. And if we have a DNA with intermediate density, so that would get at equilibrium at a point which would be lying in the midway, right? So this was the next phase of the experiment where Meselson-Stahl, they used equilibrium centrifugation method. Now the pattern which they obtained, right, the bands, the DNA bands which they obtained, they supported the presence or the existence of the semi-conservative mode of DNA replication. How? Again, this is important, right? So listen to me carefully. See, here initially when the bacteria, they were allowed to grow in the media with heavier isotope. So what do you think? The DNA that would be produced, how it would be? Whether that would be heavier or lighter? Obviously that would be heavy DNA only, okay? Because bacteria, they don't have any other option but to use the heavier isotope of nitrogen only, right? So whatever DNA would be produced, that DNA would be heavy DNA only. So here we can see that all DNA that is being located at one position only, right? So this is where we can conclude that this is all heavy DNA. As you could see that the position, it is more towards the bottom of the tube, right? Now, after the next division, or we can say in the first generation, when the DNA was extracted from the bacterial cell and it was again subjected to this method. So Meselson-Stahl found that again there was a single band, but now this band was occupying a position which was higher to the previous position, right? So that means here the density is different from the previous band. So this corresponds to the hybrid DNA, right? The density of the hybrid DNA, because we have the heavier DNA as well as the lighter DNA, because now the bacteria, they have been shifted to the media with the light isotope of nitrogen. Now, following this means as the experiment was carried further, so here they obtained two bands this time, right? Now, one band was similar to the previous band, but here you could see another new band has appeared. Now, this new band, that is corresponding to the lighter DNA, right? Because now we could see that here the daughter DNA duplexes, they are being produced, which are all light. Now, why this is so? Just have a look at the diagram. Let me know if you have any confusion on this. See, these are the hybrid DNA. Now, when they'll open up, when the unwinding would happen, you will have the heavier DNA stand as the template as well as the lighter DNA stand as the templates, right? Means both of them, they would be acting as a template, and against them, the complementary strands they would be produced, right? So you would get the hybrids as well as you will get the daughter DNA duplexes, which would be made up of both lighter strands. So that would be corresponding to this lighter band. Am I clear with this? All right. Now, if we are clear with the semi-conservative mode, if we have understood that how Meselson-Stahl experiment that was able to give or to provide evidence in support of semi-conservative mode, so let's move to our next section. And here we'll be discussing about the rules of DNA replication. So, the first rule is that DNA replication takes place in S phase of the cell cycle, right? It occurs once per cell cycle. Now, this is also important because the cells, they are dividing. So, this is really important, the DNA replication that should happen only once, or otherwise what would happen? There would appear alteration or changes, right, in the number of the chromosomes. Next rule is that the DNA replication always starts from the origin points, right? The replication doesn't start at the random points, but it always starts at some particular points which are known as origin points. And this is true for both prokaryotes as well as eukaryotes. Now, this is the most important rule which I am going to read here. This is DNA replication takes place from 5' to 3' direction. So, you need to note down this. Okay, no matter in whatever form they ask you this question, you have to abide by this rule that DNA replication always takes place in 5' to 3'. This is because the DNA polymerase enzyme, it polymerizes in 5' to 3' direction. DNA replication can be uni or can be bidirectional, but let me tell you that bidirectional is the mode which is the most favored one, right? That means it has been adopted by most of the or majority of the organisms. So now what do we mean by bidirectional or unidirectional? So look at this diagram, right? The first one, this is depicting the bidirectional replication. So starting from the origin point, if the replication forks, now these are the replication forks, right? Now, these are the basic terms which we use in replication fork or replication bubble. So, this bubble-like structure which you could see here, why it is appearing? Because the two strands of the DNA, they are opening up. Okay, so they would just look like a bubble where the DNA is opening up. You would find that a bubble-like structure that would get created. So, that is known as replication bubble. And these Y-shaped structures, right, which would appear at each end of the replication bubble, these are known as the replication forks. So if starting from the origin points, the replication forks, they are moving in both the directions, so that is known as bidirectional mode. If it would continue in just one direction, that is known as unidirectional. But unidirectional is rare. It is seen in some viruses or some organelle replication events. All right, so here comes our next question, which has been asked previously. So we have already learned this rule. So I'm expecting quick answers to come in the chat section against this question. Synthesis of DNA occurs during the dash phase of the cell cycle. Very nice, Shrivani. That was really quick. Right. So this is S phase. All right. So we got answer to this question from our rules for DNA replication section. Now let us move to our next slide. Now this is again important, right? Thing which you need to understand regarding DNA replication. I have told you about the replication bubble, right? And then replication fork. So at each end of the replication bubble, you would find the replication forks. That is correct, Pratik. Satya Narayan, Kaushal, just someone I could see your name like that. Only Niharika, Jason, Shivani. So all correct answers there. Well done. All right. So let's get back to this diagram. So here what we are finding that this is the replication fork. I always tell my students that whenever you imagine a replication bubble, you need to keep in your mind that you have two replication forks moving in both the direction, right? Unless it has been provided that this is the unidirectional mechanism which is being followed. So remember, whatever events they are occurring at one end of the replication bubble, the same would be occurring on the other side. Okay, so this is the replication fork, right? And always remember, since the two strands they are running anti-parallel, and I have already told you that replication would always take place from 5' to 3' direction. Now since the two strands they are anti-parallel, so how it would be possible for the replication to maintain a single direction, that is 5' to 3' direction, right? So definitely it means that the replication would be occurring in opposite directions to comply to this rule, right? So for the leading strand, the replication would be occurring in the direction of your moving replication fork. So follow this arrow, right? This is really important to understand that for the leading strand synthesis, you will find the arrows, they will be heading in the direction of the progressing replication fork. Whereas for the lagging strand, you would find that they would be heading opposite to it, right? And like I've told you that at each end of the replication bubble, you will have replication fork. So at each end, you will have one leading and one lagging strand, right? So I would suggest that you must practice on the drawing the application bubbles and application forks. So you would understand that how the two strands which are running anti-parallel to each other would be replicated simultaneously, and that too complying to our thumb rule, which is 5' to 3' replication. Okay. All right. So before we go ahead, I'm sure by now you have information on this, how you can get connected to us. But in case if there is someone who is new to the session, or if you have missed on this part previously, so let me give you a quick glance that what are the different ways through which you can reach us. Okay, if you have any technical query in joining to any session or downloading our app or anything, so you can drop your messages here, or you could contact us at our toll-free number. And please join BioTecNika on Telegram app. You will get all the updates regarding any upcoming webinars or sessions. Okay, so don't miss on them. Post your feedbacks, testimonials, will really appreciate. Okay, so you can drop your messages here, and don't forget to download the BioTecNika app. All right. Okay. So let's move to our next slide. And this is about the enzymes involved in DNA replication. So I'm showing you a table here. This table is very important. Like I've told you that this part is included in CSIR syllabus as well. Okay. So if you are going to understand this topic well, if you are able to memorize the names of the enzymes here only, that is going to help you, that is going to benefit you in the CSIR preparations as well. Okay, so here what do we have? The names of the enzymes. Okay, and DNA polymerase, definitely this is the most important enzyme because this is going to polymerize the DNA. That means it is going to help with the replication, right? But this is not DNA polymerase 1, which does or performs the replication in prokaryotes, but this is DNA polymerase 3. So this is another important point which you must note down if you are not aware of this. Okay. Although all the DNA polymerases, they can do it, but DNA pol 3 is the main enzyme which is responsible for performing the prokaryotic DNA replication. Now, beside that, helicases are those enzymes which will help to open up the DNA. Like I have told you that the two strands of the DNA, they have to be separated. They need to be unwind, right? So that the two templates, they could be received. Okay. Now, this would be possible because of the helicases. They will be opening up the DNA helix. They'll break down the hydrogen bonds present, right, between the bases of the two complementary strands. You could be directly asked about the names of the enzymes and their actions. So this is the reason I'm emphasizing on this table and the names of the enzymes which have been mentioned here. Okay, you can go through the table later as well, note down the names of the enzyme, memorize what functions they have to perform. Now, what is the role of ligase? Ligase, this is responsible for sealing the gaps between the Okazaki fragments. Like I've told you, that lagging strand, that will be synthesized in opposite direction to the progressing replication fork. So it is actually synthesized in the form of the small fragments which are known as Okazaki fragments. But later on, those Okazaki fragments, they need to be united, right? They need to be joined. So their DNA ligase will help. Okay. Next we have primase. So what is the role of primase? It synthesizes RNA primers. RNA primers, they are essential for the replication event. Next we have SSBs, or single strand binding protein. Even when the two strands of the DNA, they have been separated, they have a tendency to get recoiled, or maybe they get to form any unnecessary secondary structure. So to prevent that, SSBs, they bind to each of these single strands which have been generated, right, because of the action of the helicases, and that is where the SSBs, they are helpful. Now you need to know about the sliding clamps as well. Now, what are sliding clamps? I show you a diagram here. Quickly, let us have a look at how sliding clamps, they are important. Now see, DNA polymerase, this is DNA polymerase, right? DNA polymerase is the main enzyme for performing the replication. However, it has a tendency to fall off the DNA again and again. Okay, so it needs to be stabilized there. So there, the sliding clamps help. So sliding clamps, these are donut shaped structure, the circular structure as you could see here. Okay, so they would be supporting the DNA polymerase on the DNA. So that is why they have been named as sliding clamp. Clamp is something which is used to hold the other thing tightly, right? And these sliding clamps, they are loaded by the clamp loader. So that what you are finding here, these are clamp loader. Okay, so if you can remember that, that's well and good. But this would be sufficient if you are able to memorize the names of the important enzymes, right? Means other than clamp loader, the other enzymes which we have discussed earlier. Now, topoisomerases, this is DNA gyrase, right? And topoisomerases, they help to relax the stress which builds up when the double stranded DNA, it opens up. DNA gyrase is the topoisomerase which is used in prokaryotic or E. coli replication event. So do we have any doubt on this? So I hope you would note down all the names with their functions from this table. Okay, so let us quickly look at this question here. DNA gyrase is required for. So we have just discussed that for what DNA gyrase would be used for prokaryotic DNA application. So yes, can I get the answers in the chat section? Okay, so here come the answers. Jason 4, Niharika 4. All right. So I hope this is clear for everyone, right? Relaxation of DNA. So the main purpose in the DNA replication which is being targeted by DNA gyrase, which is a topoisomerase, is the relaxation. Relaxation in the sense the stress which would build up, that needs to be relaxed. Okay. And stress would get generated because we are opening up the two tightly coiled DNA strands. Now, the next question is, polymerization of Okazaki fragments during replication is mediated by? Yes. So, which of these enzymes is going to help with the polymerization of Okazaki fragments? Okay, Jaypreet Singh says uh, 2. Purvi, this is 1. All right. Not exactly. Okay, let me tell you, this is DNA polymerase 1. Okay. See, helicase, that would help to separate. DNA polymerase 3, that is going to polymerize, right? Means extend the DNA during replication. DNA polymerase 2, this is mainly required in repair. So DNA polymerase 1, that this is the enzyme which would be helping with the polymerization of Okazaki fragments, or we can say that filling up of the gaps, right? When the Okazaki fragments, they would be joined later on. Okay. And each of the Okazaki fragment, that would be having its own primer. See, this is one primer and this is one Okazaki fragment. This is another primer, this is another Okazaki fragment. This is another primer for the next Okazaki fragments. Now, when these primers, they are taken out later on, so you can imagine that the space, right, there would be voids, right, vacant spaces which would be created in between these Okazaki fragments. So these spaces, they would be filled by DNA polymerase 1. Okay, and finally DNA ligase will seal the nick. Okay, so this is how DNA polymerase 1 is involved in DNA replication. Okay, it doesn't perform the main replication event, but it has a major role to perform there. Now, next we are going to discuss on the Chargaff rule. This is another important thing, or important rule, I would say, right? Based on this, the questions had been asked previously. So what is Chargaff rule? Let us quickly see, right, what this rule states. According to this rule, total number of purines in DNA are always equal to total number of pyrimidines. So I hope you know that what are purines or what are pyrimidines. Okay, so purines, adenine and guanine, they would be equal to thymine plus cytosine. Okay, why this is so? Because adenine, that always base pairs with thymine, and guanine, that always base pairs with cytosine. Right? Now, based on this, let us attempt these question. In fact, the first one, right? This is exactly based on the Chargaff rule. Yes. So this question asks, if the ratio A plus G by T plus C of one strand of double stranded DNA is 0.6, then what is the ratio for the same in the complementary strand? Yes. I would like to have quick answers on that. I hope you understood the Chargaff rule. All right. Okay. Kaushal 1.4, Shivani 0.6, Jaypreet 4. So how you are solving it? What concept you are using here? See, remember, in one strand of the DNA, if we have A plus G by T plus C as 0.6, okay, so in the complementary strand, you will have inverse of 0.6, right? So in one strand of the DNA, you would have A plus G by T plus C, right? Now, in the complementary strand, whatever amount of A plus G you are having, you will have T plus C, because adenine, that is going to base pair with T, right? G, that is going to base pair with cytosine. And similarly, we can say for thymine and cytosine. So whatever ratio you are having or you have been provided here, you need to calculate the inverse of it. All right, now try solving it. And don't forget to leave your answers in the comment section. Although you are going to find the answers uh, in the free notes, right, which you can download later, but I would like to see your answers coming in the comment section as well. Okay, so I hope you got to understand, right, that what concept actually we are applying here. Now, the next question is, the stability of a DNA is determined by? Very good. Just someone, I could see that you have already got the right answer. Yes, that is correct. Kaushal, Rinki, Pal, you have mentioned option 1. So I'm not sure for which question you have pulled in this option. Okay, but this would be number of cytosine and guanine bonds. So remember, GC base pairing being more stronger, right? So this marks more stability for the DNA, right? So whatever DNA means, the DNA belonging to a particular species, if it is rich in GC content, so that is considered to be more stable. Okay, so yeah, yes, that is correct, Muhammad Rauf, option 3. Okay, now let us move to our next slide, and let me remind you, all these questions they have been asked previously. Okay, so now I'm sure you are getting an idea that how they are related to these topics, what all topics you need to focus at, so that you could be able to solve the questions asked in the exam. So here I'm going to give you a glance that how the DNA is extended during DNA replication. See, this is the template strand, right? And as you could see that these are the nitrogen bases, and this is the new strand which is under synthesis. So here you would find that a new nucleotide that would appear each time, and the reaction, the phosphodiester bond formation reaction, that would be catalyzed by DNA polymerase, and this new nucleotide will become part of the extending DNA chain. Okay, so you could see that this is the phosphodiester bond which has been formed. All right, so that is how the DNA polymerase helps with the extension of the DNA. And even before that, let me tell you that this is ensured that the base which is being carried by the incoming nucleotide, that should be correct as per the base present on the template strand. Okay, so here the hydrogen bonding will take place first, and then the phosphodiester bond formation would be done by the DNA polymerase. So this is the same thing which has been shown in the other form. Okay, so I hope you are finding these diagrams helpful. So here again you could see the nucleotide is coming, and this is being added to the growing DNA chain. Now, this slide is again very important. So please look carefully at these tables, right, which I am sharing here. So this is regarding the DNA polymerases for prokaryotes, right? We have separate or a different set of DNA polymerases for prokaryotes and eukaryotes. Okay, so this is the information regarding DNA polymerases in prokaryotes. So here you could find all the information, right? The main DNA polymerases which are present in the prokaryotes. Then let me tell you that whatever DNA polymerase it is, all of them they are going to polymerize in 5' to 3' direction only. However, they show exonuclease property, right? Exonuclease property means they can degrade the nucleotides, right? So this is just the way opposite to polymerization. Now, some of the DNA polymerases they may show 3' to 5', the others may show 5' to 3'. Okay, and DNA polymerase 1 is the enzyme which shows both of them, right? For most of the others, you would find that 3' to 5' exonuclease property is available. But DNA polymerase 1, it has both these properties. So this is one point which you must note down, right? Now, the next column, you are going to find the functions, right? The functions which these DNA polymerases perform during DNA replication. So this is another important column, right, which you must go through. Right. So this is another table which is again about the DNA polymerases for prokaryotes, and particularly DNA pol 1, pol 2, and pol 3. Right. Now you may ask that although all the DNA polymerases, they do have the polymerization ability, that too in 5' to 3'. So why it is so that DNA polymerase 3 is being used and not the other DNA polymerases? So here is the answer. You would find that the polymerization rate for DNA pol 3, this is the maximum. Okay, why it is so? Because it has the maximum processivity. Now, what do we mean by processivity? Nucleotides added before the polymerase dissociates. So I told you that the polymerase, it has a tendency to fall off the DNA replication. Okay, so whatever number of nucleotides it can add before it falls off or it dissociates, that is known as processivity. So this remains maximum or highest for DNA pol 3. This makes DNA pol 3 to be used as the main replicative polymerase. Okay, and this is about the eukaryotic DNA polymerases. So these are the eukaryotic DNA polymerases. Again, this table is important. You could be directly asked about the functions, the names, and then their corresponding functions, right, in the eukaryotic replication unit. Now, let's move to our next slide, and here we are going to learn another important term, which is clenofragment. Right. Now, what is clenofragment? This is actually obtained from DNA polymerase 1 only. Right. DNA polymerase 1. This exists as a holoenzyme structure. Means we can find different subunits, right, or different domains in the DNA polymerase 1 structure. And as I have told you that DNA pol 1 is an enzyme which is having both types of exonuclease activity. So here you would find that there is one domain which is showing the polymerization property, and there are other two domains which are showing the exonuclease property. Now, when DNA polymerase 1, this is subjected to proteolysis, right? So it dissociates into two fragments, the larger fragment and the smaller fragment. The larger fragment, that is known as the clenofragment. So you could be directly asked that what is clenofragment, right? So you need to know that this is a fragment actually which is obtained from DNA polymerase when this is subjected to proteolysis. Okay, to the protease treatment, subtilisin treatment. So here this clenofragment that would be having 5' to 3' polymerization property and 3' to 5' exonuclease property, right? So it will not be having 5' to 3' exonuclease property anymore, right? So this what you need to note down. What is clenofragment and what it all is having, what properties it is having. So it has a great use in the molecular biology experiments, right? Using these properties, clenofragment is used in designing the molecular biology research experiments. All right, now come to another important name, which is telomerase. Right now, before knowing telomerase, you must know that what are telomeres. See, in eukaryotes, now we know that eukaryotes, they do have linear chromosomes, right? So there comes an issue, there comes a problem where the DNA replication, it cannot be completed at the ends of the chromosomes. Okay, and because of that, with every replication cycle, some part of the chromosome that would be lost. Okay, so just imagine if it would be happening with every cell division, with every replication cycle, so some important sequences, they could be shed off from the ends of the chromosomes. So perhaps to avoid that, the nature, it has provided the ends of the chromosome with some repetitive regions, right, which do not have any genetic significance as such, and those sequences, they are known as telomeres. So telomeres, these sequences, they are repetitive sequences present at the ends of the chromosomes, I repeat, and they protect the chromosome from degradation or fusion with neighboring chromosomes. And not only that, they would be helping with the maintenance of the chromosome length. Okay, now vertebrate telomeric sequences TTAGGG. So you need to note down this. It has been asked previously. All right. So here, telomerase, this is an RNA dependent DNA polymerase that synthesizes telomeric DNA sequence. Okay. Now it's a reverse transcriptase enzyme. This is important. Please note down this property of telomerase. This is a reverse transcriptase enzyme. You understand what do we mean by reverse transcriptase? Yes, it is able to synthesize DNA against RNA. Okay, and it would be containing its own RNA template. Means it carries the RNA template with itself. In vertebrates, the sequence is this. So you would find that this sequence is actually complementary to the telomeric sequence. Telomeric sequence is the one which you are going to find at the ends of the chromosome. And this is the sequence which you are going to find in the RNA template being carried by the reverse transcriptase enzyme, which is telomerase. So it helps to maintain the ends of the chromosome. However, it doesn't work in the normal somatic cells. Okay. This is active in normal stem cells only, and if the telomerase becomes over functional, so there the somatic cells, they may turn cancerous. So let us have a look at few questions, right, which are related to the telomeres or the telomerase concept. Okay, so our first question is, telomere repeat sequence in humans is? Very good, that was really quick. Very good, Kaushal. So that means you are really attentive during the session. Well done. So now our next question, which of the following enzymes has reverse transcriptase activity? So see, these questions, they had been asked earlier. So they are related to what we have discussed on our previous slide. Very nice, Prateek, Purvi, Jason, Shivani. I really appreciate that all of you are trying on solving these questions. So I hope you are finding them easy now. When we have discussed the concept related to them, that would help you to retain these topics for longer. Am I right? Yes. So definitely this is telomerase. Okay, telomerase is having the reverse transcriptase activity. Remember, telomerase that is having RNA template as part of a structure only, which it would be using as the template, and this would be synthesizing the DNA to extend the ends of the chromosome. But like I've told you, that this won't work in the somatic cells. Okay, now let us move to our next section, which is DNA repair. DNA repair, this is the name given to the collection of the processes, right, which are important to maintain the integrity of the genome and hence to maintain the functionality of the individual. Okay, we all are vulnerable to DNA damages, right, which may be generated because of the exogenous sources or maybe because of the internal causes. That means the endogenous sources. So that makes DNA repair really important to work, so that the DNA damages, they could be fixed, right, and the integrity of the DNA, that could be maintained, right. Just read out the statement, right. So DNA in a human cell that undergoes several thousand to a million damaging events per day. So just imagine if these damaging events, they won't be fixed. So what would happen? The mutation, they could be caused in the DNA. So the transcription, the translation processes, they could get affected, right. And if these mutations, they are inherited, so that would again be something which would be dangerous, right, which would be lethal to the existence of the human beings, right, or any other species organisms, right. So here mutations is equal to DNA damage minus DNA repair. If due to any reason DNA repair mechanism, they are not working efficiently, so the mutations, they would arise. Now here, what do we have on the slide? This is about the causes and the types of the DNA damages. Okay. So this diagram would give you an overview that how different DNA damages, they could be caused, right? What could be the reasons? So like I've told you, that cellular metabolism means the intrinsic reason. Do you know that the heat which is generated when the metabolic reaction, they are taking place in our body, that can cause uh, the bases to get removed from the DNA. Okay, so depurination or depyrimidation may happen, means a purine may go missing or a pyrimidine may go missing. Okay, and apart from that, replication errors. So although the replication, that is quite an efficient mechanism, but even then there are chances that replication errors may happen, and because of that, the double strand or maybe single strand breaks, they could be generated. Ionizing radiations like cosmic rays or the radiations which would appear because of radioactive decay, so they can cause single strand breaks or double strand breaks to appear. Ultraviolet radiations, right, they may give rise to thymine dimerization. That is the most commonly found effect because of the UV light exposure. Then toxic chemicals, all the industrial effluents, all the toxins which are existing in the environment. Okay, so they may cause maybe alkylation, oxidation, deamination of the bases, right, which are integral to the DNA structure, and there appear the DNA damages. Okay, so these are the types of DNA damages which may appear. So see, single strand break, right? Double strand break, where you would find both the strands of the DNA, they have been broken. Damaged base means the base that could be damaged, it could be modified, like I've told you. Alkylation may happen, like vinyl chloride, right? That is a common example I can give. Intra-strand cross-link, right? So within the strand, you would find the cross-link that has appeared. Then interstrand crosslinks. Okay, so there it becomes important that DNA repair mechanism should work, right? So before that, checkpoint activation would happen. Okay, so this you will learn under a different topic, right? If you have learned cell cycle, so you must be having an idea on that. Now let me tell you that if the DNA damages, they are crossing the threshold limits, so the cell may be sent for apoptosis, that is programmed cell death. So I'll explain this topic in my next session. So that would also remain an interesting session. I would request everyone to join in that as well. Okay. So these are the different possibilities which would exist to fix the DNA damages. Okay. Now here you need to learn the three categories into which we can keep the DNA repair mechanism. So I'm giving you this flow chart. This would help you to remember these three categories here. The first one is the direct reversal. So here, basically we have two DNA repair mechanism: photo reactivation and methyl group removal. Then the second one is the single strand damage repair mechanisms. So these are the three names which you need to memorize: base excision repair, nucleotide excision repair, mismatch repair. Okay, so you need to remember that these work to fix the single strand damages. Then we have double strand break repair methods. Okay, so here again we have three names: non-homologous end joining, homology directed repair, microhomology mediated end joining. So at this level, you don't need to go into the details. However, when you'll be preparing for CSIR or the other examinations, so there you would be required to learn all the details, the names of all the enzymes and the factors which are involved in these mechanisms. Now, let us look at another question here, which asks, which of the following is not needed for DNA transcription? So that indicates now we are moving to our next section, which is DNA transcription. Yes, so what would be the answer here? Yes, quickly, can I get answer for this question? Which of the following is not needed for DNA transcription? Okay, let me reveal the answer here. This would be option 1. Why won't the nucleotides be required for the transcription? Nucleotides, they would be required, right? Because the RNA that would be synthesized, but we don't need ribosomes in transcription. Okay, so they would be required during translation. So those who are not clear with that, what these terms mean, transcription or translation, so let me tell you that, see, DNA that can replicate, okay, so that means it can form duplicate copies of itself. Apart from that, it can transmit the information to RNA. Okay, that means RNA would be synthesized against the DNA template. This is known as transcription. Okay, so here the ribosomes, they are not needed. All right. But when the information would be further passed to the proteins, so that process is known as translation. Right, that means translating the information further in the form of the protein. So there we would require the ribosomes. Okay, so I hope that would help you to remember these terms later as well. Now let us learn more about the transcription. So synthesis of RNA molecules using DNA strands as the templates. Now Let me tell you that usually it remains as a confusion among students that they think that mRNA is synthesized through transcription. That is correct. Yes, mRNA is synthesized means the information that would be passed from DNA to mRNA and then to proteins. But that doesn't mean that the other forms of RNA, they are not being produced through transcription. Okay, so besides mRNA, which is the coding form of RNA, the other forms of RNA, let it be rRNA, right, which would be used for the formation of ribosomes. Okay, rRNA that joins, that associates with the proteins to produce the ribosomes. Then tRNA, then other small forms of RNA, snRNA, snORNA, or small cytoplasmic RNA. Okay, so they have various roles to play in the cell, in the cellular processes. So all these types of RNAs, they are synthesized through transcription only. Okay, now to learn more about transcription, so this is a diagram which is going to give you an overview, right? We firstly need to visualize, we need to imagine how the transcription is happening. So have a look here. This is the double stranded DNA structure only. Now what we are looking, that again DNA is opening up, and this time the enzyme which is involved in the synthesis of the RNA against the DNA template is RNA polymerase. Makes sense, right? Since this is synthesizing RNA, so this would be known as RNA polymerase, not the DNA polymerase anymore, right? So I hope this is clear for everyone. Okay, the stretch of the DNA transcribed into an RNA molecule is called a transcription unit, and it is going to encode at least one gene. One gene that would be at least encoded. More than one gene could be there, but that happens in case of prokaryotes only. If you know about operons, so operons, you know that there we can have more than one gene which would be coded, right, under a single transcriptional event. Somewhere we have 3, somewhere we have 5. Okay, but in eukaryotes you will find that only one gene that would be encoded during one transcription event. So what we are looking here, this is RNA polymerase, right? This is continued on DNA using it as the template, and this is your RNA, right, which is being produced. It has been shown in green color. I hope this is visible to everyone. Now, another point which you need to extract from this diagram and from this slide is that RNA is being transcribed in 5' to 3' direction. Okay, so you need to note down whether this is replication, whether this is transcription. The synthesis is being taking place in 5' to 3'. So don't get confused if they give you replication or if they give you transcription. So in which direction it is, or the synthesis is being taking place, this is 5' to 3'. So this is where you could see the nascent RNA molecule. Nascent means this is still needs to be modified. Okay, it would be sent for the processing. That too particularly in case of eukaryotes. Okay. Yeah, that's correct, Shrivani. Very good. Monocystonic and polycystonic. Good to see that you are aware about these terms. Okay. So polycystonic in prokaryotes and monocystonic in eukaryotes. Now, so here we need to learn the terms which are used for the strand which is being coded, right? That means against which the RNA is being synthesized. You need to know that what is the term which is being used for it, and then what are the terms which could be used for the other DNA strand. Now, this is again important to be understood. See, in DNA replication, we have learned that the two strands, they would get separated and both of them they'll be treated as template. However, here you would find that only one strand that would be read as template. Okay, so this green one, this is your RNA, and the gray one, this is the template strand, the DNA template strand, which is being read for the RNA synthesis. So this is easy to remember. This would be named as template. The other would be named as non-template. Okay, I hope this is easy for everyone. But now come the other terms, right? Now these terms, they might be confusing. So you need to note down on these. Template strand, this is also known as antisense strand, right? And the non-template strand, that is known as sense strand or the coding strand. Okay, I'm repeating on this. Template as antisense, and the other DNA strand or the non-template strand, that is known as sense or the coding strand. So here you could see that if we have a DNA strand with a particular sequence as coding stand, and this is the antisense or the template stand. So this is your non-template, okay, and this is the template stand. So non-template, this is sense or coding, template is your antisense. So this is the RNA which is being produced against it. So you would realize that this would be exactly complementary to it, and because of that this will be exactly similar to coding stand, however with one major difference, that here we'll have uracil in place of thymine. So this is DNA, this is RNA, right? So the coding stand, this is exactly like the nascent RNA stand, because both of them, they are complementary to this DNA stand, but with this one difference. Okay, so this would be the difference. So I hope you have understood. Right, now based on this, let us try these questions here. mRNAs are encoded by the DNA sequences. What DNA sequence would behave as a coding stand for mRNA molecule with the sequence of the one which has been provided in the question? Yes. So whenever you get questions like this, observe that where do you have the 5' and 3' ends carefully. Okay, don't miss on the 5' and the 3' ends. This is important. Yes, Shivani, Jaypreet, Jason, Janvi, Mohammed, Kaushal. So I would like everyone to try on this. Yes, what would be the answer? Shall I reveal the answer here? Okay. So this would be option 1. Why it is so? I hope you got the question. What this question is asking: what DNA sequence would behave as the coding stand for the mRNA molecule? So this is your nascent RNA sequence which has been given, and on the previous slide only I have shown you that how the coding stand and the nascent RNA stand, they are similar, okay, but only with one difference, that you would have uracil in RNA but thymine in the coding stand of the DNA. Okay, just someone, it won't be option 3. Okay, all right, even if that is so, Purvi, so we would be considering both of them as correct. Okay, because we need to pay attention to that, where we have the labels, and then how this is being complementary to the RNA nascent stand. Okay, all right, but one with one difference, that will have uracil, right, in place of thymine in the nascent RNA stand. All right, now let us solve our next question. Which of the following is the base sequence of the complementary strand of double helical DNA in which one strand has the sequence this one? So see, now look at the question carefully, right? This question was about the mRNA, the coding stand for mRNA. Here you have been asked about the double helical DNA. Very nice, Kaushal. That was quick. Okay, so this question is really simple, right? This is just asking the complementary stand for the given DNA stand. But again, one thing which you should be careful about are the ends. Okay, you have to be careful that whenever you are choosing one option, you are making your option, you are making your choice there, so you have to mind it that wherever you have the 5' end in the template stand, you will have 3' in the daughter or the complementary stand, because they would be opposite to each other, they would be complementary to each other but in opposite orientation. Okay, yes, Shivani, that is correct. Very good, Purvi. Okay, now let us move to our next slide, and this is about the RNA polymerase which works in the bacteria. Right, now here the important information which you need to extract, which I need to focus, is about the sigma factor. Now what is sigma factor? RNA polymerase, again this is a holoenzyme structure in bacteria, means this is made up of different subunits. As you could see here, this is having 2 alpha subunits, right, which are exactly identical, then similar but not identical beta and beta prime, and omega subunit which is not visible here in this diagram, but yes, it is there, right, as part of the RNA polymerase whole enzyme structure. And then beta. Beta is the catalytic subunit. But apart from this, the sigma factor is really important. Now, why this is important? Because without the sigma factor, the core RNA polymerase, I mean, whatever units which have been left, apart from sigma factor, they make up the core RNA polymerase, right? They have the potential to associate with the DNA, right, when they have to perform the transcription. However, the core enzyme that would bind to any random location, right, until and unless sigma binds to it, right, sigma binds to the core polymerase, and then this sigma factor helps this holoenzyme to locate specifically to the promoter. So I hope you know that promoters, these are the sequences over the DNA where RNA polymerases bind. Okay, and who is helping the RNA polymerase to locate the promoter specifically? This is sigma factor. All right, so I hope you have taken down this point. There should be no confusion. Shall I repeat? Sigma factor, this would be binding to the core RNA polymerase to form RNA polymerase holoenzyme structure. Only then this structure would be able to identify the promoter specifically, and it would bind there to form the pre-initiation complex. Okay, and following that, the transcription that would be initiated. Now, since we are talking about the promoters, so let us know more about the promoters here. And here you need to take down another important name. This is Pribnow box. All right, now what is Pribnow box? This is the name given to a specific sequence which is located at -10 position to the TSS or transcription start site. Okay, so the positions which lie upstream to the transcription start site, they are designated with minus sign. Okay, so here you would find a specific sequence is present. Can you see this? TATAAT. Okay, so this is known as Pribnow box. Now, apart from that, there remains another sequence. This is again a consensus sequence, -35. All right. Okay, so both of these sequences, they would be helping the RNA polymerase in bacteria, right, to bind, and then the double-stranded DNA would open up in or around the promoter region. So these are acting as the promoters. Tata box, Shivani, Jason, Tata box, that is a eukaryotic promoter. Okay, so yes, that is something similar to the Pribnow box. So you can say that Pribnow box, this is the promoter for bacteria. Tata box, that is the promoter for eukaryotes. Right? So that is the difference, Jason and Shrivan. Okay, so you need to remember this term Pribnow box. You need to remember the sequence as well. You need to remember that this is located at -10th position, as well as you need to remember that this is the bacterial promoter. Okay, so this is how the transcription would look now when the transcription bubble would appear. So see, this is your Pribnow box, -10 position. This is -35 position. So these act as the promoters. So RNA polymerase is going to bind, right, somewhere like this. Okay, that would be occupying these promoter sites, and the DNA would open up. Okay, DNA would open up near the promoter site, as you could see here, and this is known as transcription bubble. Right, now within the transcription bubble you would find that TSS would be there. TSS means transcription start site. So where we have the transcription start point here, and this is what, this is the RNA which is being synthesized. Okay, remember RNA would be synthesized in 5' to 3' direction. This is how it would be extended. All right, so keep this diagram in your mind when you need to visualize the transcription to solve any question based on the transcription. So here comes a question which has been asked previously based on the transcription concept. See, now you will find it quite easy. I expect everyone to answer. Tata box and Pribnow box, these are what? So the answer is already there. Yes, these are promoters. So Tata box, this is the eukaryotic promoter, like I have mentioned earlier. Pribnow box, this is the prokaryotic promoter. Clear to everyone? So I hope there should be no doubt. If you have any doubt, you may ask in the chat section. Okay. Now let us move to our next section, which is RNA processing, post-transcriptional processing. So yes, this is really important in case of eukaryotes, right? The RNA which would be produced, that would be sent for processing, right? So DNA, that would be transcribing to RNA, and then different forms of RNA, they would be produced. All of them, they would be going under some type of processing. However, RNA processing, that remains most extensive for the mRNA, and that too in case of eukaryotes. Now, why this is so? Why RNA processing extensively happens for eukaryotes, not for prokaryotes? Now, this is very important question, right, which could be asked. This is because in eukaryotes, the transcription and translation takes place in different compartments, right? Transcription takes place in nucleus, translation takes place in cytoplasm. However, in prokaryotes, both of them, they occur in cytoplasm only. Okay, so the RNA that is being synthesized there in the cytoplasm only, and then it would be sent for the translation. See, now what do we have here on this slide? DNA transcribing the RNA. RNA is known as primary transcript or nascent transcript, and then this is further going for translation. So this is the protein which has been produced. This is in case of prokaryotes. So both the processes, they are occurring at a common location, that is cytoplasm. So RNA, that won't be sent for processing. There is no time for processing, right? However, in case of eukaryotes, the transcription, that is occurring in nucleus. So you would find that RNA that is being synthesized in nucleus, then it has to travel out of the nucleus to reach the cytoplasm for undergoing translation. So there it needs to be modified. Okay, it needs to be processed. So that is known as processing, RNA processing. Yes, that is correct, Shivani. Good. So now what are the processing events which are found in the eukaryotic mRNA processing? 5' capping, 3' cleavage, polyadenylation, RNA splicing, RNA additive. Okay, so you need to remember all of these 4 processing events which are important to the eukaryotic mRNA process. So here you could see that this is the RNA which is being produced. And now another thing which you need to know and to remember is that capping will be done at the 5' end. This is really important, right? You could be directly asked that at which end of the RNA the capping is done or the polyadenylation is done. So as you could see here, capping is being done at the 5'. Okay. Whereas the polyadenylation, the poly A tail attachment, that would be done at the 3'. Right. Good, Shivani. Okay. So nice, Atanu, Samantha. I could see that you have also pulled in the right answer for my question, that why the processing is required particularly for eukaryotes. Okay, now capping and polyadenylation at 5' and 3' end. Apart from that, splicing. So splicing is where the introns, they would be removed and exons, they would be joined together. So then only the final mRNA that would be produced, which would be translated. Okay, now somewhere the RNA editing can also be done, where some bases, they would be modified intentionally under RNA editing mechanism, right, to bring diversity in the eukaryotic protein pool. All right, so our next question. Post-transcription processing of mRNA is a routine process inside the cell in order to complete the translation process. A gene of the mature mRNA will not have the following region. Yes, so what do you think? Which of the following will not be present in the mature mRNA? So that what question is asking. Again, this has been asked earlier. Yes, that is correct, Tanu. Kaushal. Yes. So can I get answers for this question as well? Quickly, we need to maintain our pace here because we are still left with another section, which is translation. Yes, that is correct, Jason. Very good. So that would be promoter, right? Promoter is present in the DNA. So once the transcription has happened, now we have been provided that this is the mature mRNA, so it will not have the promoter. Okay. Now let us move to our next section, which is about the protein synthesis, that is translation. So see, we have covered a long journey, right? Starting from DNA, then the replication of DNA, then transmitting the information to RNA. RNA has been sent for processing. Now it's time for the protein synthesis, that is translation. So this is the overview, right? Those who don't know much about the translation, this diagram would help you to understand that where protein synthesis is actually placed, right, when we need to connect it with the transcription, right? So here we have the DNA, this is the RNA polymerase, so mRNA that is being synthesized. Okay, so for convenience only this has been shown that all of them, they are coming from a single DNA, but obviously that won't be there. We have specific sequences which would be generating the coding mRNA or tRNAs or RNAs. Okay, so now the rRNA, they will unite with the ribosomal proteins to form ribosomes. So these are ribosomes. Okay, and mRNA, ribosomes, as well as the tRNAs. Now apart from that, the factors, okay, they will all be coming together to uh, perform translation. Okay, and the protein synthesis would take place. Okay, now genetic code. Now this is again important. You could be asked questions based on the genetic code concept. Now what is genetic code? Genetic code consists of 64 codons and the amino acid being specified with these codons. So we know that three consecutive nucleotides, they would be coding for one amino acid. Okay, and we have 20 amino acids, right, which are being coded by 64 codons. See, three nucleotides, right, which would be coding for one amino acid, and at each of the three positions there is a possibility for any of the four nucleotides to come. So by applying the permutations, so you will get 64 codons, right, like this is where we are getting 64, right, as the possibility for the total number of codons being produced. But if you have realized that we have only 20 amino acids and then 64 codons, okay, so that means we have more information in the genetic codes as compared to the amino acids which are being specified. So that property is known as degeneracy, where more than one codon that code for an amino acid. Okay, so you will find that there could be more than one codon, right? Like here you could see for serine we have 6. Okay, so like this there could be 2, there could be 3, more than one codon which could be coding for one amino acid. So that is known as the degeneracy property. This is important. Okay, so please highlight it, please note down. Okay, now apart from that, you need to remember that which codons they would be acting as the stop codons. Okay, UAA, UAG, UGA, again this could be directly asked, and the start codon, which is AUG, which would be coding for methionine. Okay, so all these points you must note down, and this is another important table which I am going to share with you, and this is about the exceptions to the universal genetic code. Yes, there exist some exceptions to the genetic code. See, here in the bacterial DNA, mycoplasma, so here what do we have? UGA, that is a universal stop codon. But in this bacteria, this acts as a codon for tryptophan. And this part is important. Okay, mitochondrial DNA, human mitochondrial DNA, here UGA, which should act as the stop codon, this would again be coding for tryptophan. Okay, like this AUA, which should be for isoleucine, but this will be coding for methionine. Okay, so this is the table which you should refer to know about the exceptions to the genetic code. Yes, Samantha, that is correct. That one codon that can code for only one specific amino acid, that is correct. But what I am saying is that one amino acid that could be coded by more than one codon, like I have shown you here. So all these codon, they are coding for serine. Okay, so they all are known as synonyms, right? Just like we have the words which mean same in English, they are known as synonyms. So all these codons, they would be coding for serine. This is specific that this will always be coding for serine. This again will be coding for serine only, but not any other amino acid. But what we are trying to understand here, that one amino acid that could be coded by more than one codon. This is the reason that we have 64 codons which are coding for only 20 amino acids. Okay, so I hope you understood that point. Now, this is how we can imagine the translation process to happening. Protein synthesis can be divided into 4 stages, right? The first is the binding of amino acid to the tRNA, right? So this is a point which we are usually not aware of. We know about the initiation, we know about the elongation and the termination. These are the other 3 stages which happen during the translation. So now what do we have here? This is mRNA, this is the ribosome, this is the start codon. So a tRNA would come that would bring the corresponding amino acid as per the codon on the mRNA, okay, and that is how the protein synthesis that would happen. So that would read the information which is existing on the mRNA. That is known as translation. But even before that, let me tell you that tRNA, they bind to the amino acid. Only then they'll be bringing in the right amino acid. Yes? Okay. So that is important. Okay. That is an important event. We must understand this as well. So here, this is about the tRNA structure. Clover leaf-like structure, the secondary structure. Okay, so this could also be asked. So you must have an idea on the basic structure of the tRNA. It has different loops. Okay, so this is pseudouridine loop, this is anticodon loop, this is dihydrouridine loop, and the most important part is the acceptor stem. So acceptor stem is that part where the amino acid will get attached to the tRNA. So what we are looking here, this is actually the RNA, right? This is the 5' and this is the 3' end of the RNA. And it has formed this secondary structure where some portion they'll get into base pair. Okay, so that is how the secondary structure of the tRNA that is being produced. Okay, and like I told you, acceptor stem, this is important. And at the acceptor stem, we have a stretch, right? CCA stretch. And this exists in 5' to 3' orientation. Okay, so you need to remember that the amino acid that is attached to the 3' end of the tRNA. Okay, so this is the adenine, right? From this stretch only existing at the 3' end, and what we are looking, that amino acid that is being attached. So I repeat on this. This is tRNA with different loops. This is the acceptor stem which has the stretch 5' to 3' CCA stretch, and at the adenine on the 3' end, the amino acid that would be added. So this is known as tRNA charging. When the right amino acid that is attached to the tRNA, this is known as tRNA charging. So remember, when we say charged tRNA means the tRNA that has been attached to the right amino acid. So this takes place with the help of the enzyme aminoacyl synthetase. Okay, that is the enzyme responsible for adding up the right amino acid to the tRNA. So how this process takes place? So you need to remember the basic reaction only. See, this is the amino acid. Okay, so in the very first step, the amino acid that would react with ATP. And this is known as adenylation, where the AMP that would get attached to the amino acid. I repeat, this is amino acid, ATP is being used, and AMP is being attached to the amino acid. This is known as adenylation. Yes, that is correct, Atanu. Very good. Activation of the amino acid. Okay. So now comes tRNA, right? So in the second step, the amino acid that is transferred to the tRNA, and where it would be getting attached to the tRNA. Yes, let me see who all are attentive. Can you tell me? On the previous slide only we have discussed that where the amino acid would get attached. Yes, so this is what we are looking, is the charging of the tRNA. Now this tRNA would be known as charged tRNA. So tRNA, they are specific to their amino acid. So they would be charged with the correct amino acids. Okay, and their amino acyl synthetase, that enzyme has an important role to play, which we often miss, right? We know mRNA is taking part, we know tRNA taking part, we know ribosomes, they are the one of the main components, but amino acyl synthetase, that is another important component because it is helping to attach the right amino acid to the tRNA. And that is correct, Muhammad. Very good. Acceptor stem. Okay, that is correct. So adenine at the acceptor stems. Okay, so you need to remember this stretch. All right. So yes, here we have two questions which are based on translation, which had been asked earlier. So yes, the first question: codon anticodon interaction that is stabilized by. So do you know about the codon anticodon? Do you know where anticodon is existing? We have seen in the diagram for the tRNA. Very nice, Kaushal. That was very quick. That is the right answer. Well done. And the next question. In cytoplasm, the synthesis of protein at ribosomes is called. See how easy this is, right? In fact, we have learned it in much detail now, and you have been asked that in cytoplasm, synthesis of protein at ribosomes is known as translation. Okay, so you need to focus on all the components which are taking part in translation, right? How the information is flowing from the DNA till proteins, what all are the components. So I have already shared some diagrams which would help you to visualize these important cellular processes which are taking place in the cell. Okay, so here I would like to end session for today, but we'll meet up in our next session. If you like the session, please don't forget to like it and to leave your comment. Okay, let us know that how we can help you further, right? And I had asked one question, if you remember, based on Chargaff rule. So if you know the answer, drop your answers there. And remember, knowing is not enough, we must apply. Wishing is not enough, we must execute. So execute your preparation strategy now only and connect with us. So I have already shared different ways through which we can connect. All right, then. All the best to everyone. See you in the next session. Take care. Good night. And don't forget to fill the form as well to download the free notes. So the link has already been shared. Thank you, everyone.